Alcohol, known scientifically as ethanol, is one of the simplest organic compounds that can dramatically alter the way the human body and mind function. It is made from a handful of carbon and hydrogen atoms with a single oxygen, forming a small and rather unassuming structure. Yet this simple chemical carries the power to shift perception, change mood, weaken coordination, and even alter consciousness entirely. Because of its small size and chemical makeup, ethanol can travel easily through the body's natural barriers. It slips through membranes and crosses into places many other, bulkier molecules struggle to reach. Once inside, it interacts with countless systems, weaving its effects into both body and brain, giving rise to what we recognize as intoxication. But the pathway to drunkenness is neither straightforward nor identical from one person to another. The same drink that leaves one individual relaxed and sociable might send another into drowsiness, aggression, or sadness. To understand why alcohol's influence feels so different across people, we need to trace its entire path from the moment it enters the body until it is eventually broken down and eliminated. The story begins the instant alcohol is consumed. Whether in the form of wine, beer, or spirits, ethanol first makes contact with the stomach. From here, its next destination is the bloodstream, the highway through which it will reach every tissue. The stomach lining absorbs a small portion directly, but the majority of absorption happens farther along in the digestive system. The small intestine, with its enormous surface area and rich blood supply, is especially efficient at pulling alcohol molecules into circulation. Once swallowed, the speed of alcohol's entry into the blood depends heavily on what else has been consumed. If a meal has just been eaten, the stomach is full and digestion is slower. A muscular valve called the pyloric sphincter closes when food is present, preventing the contents of the stomach from rushing into the small intestine. Because alcohol must wait until food is processed before moving forward, its absorption slows dramatically. Drinking on an empty stomach, however, allows ethanol to move quickly into the small intestine, where it is absorbed at a far greater rate. The difference is striking. The exact same drink taken after a large dinner may raise blood alcohol levels only a fraction compared to the same drink consumed without food. As alcohol crosses into the bloodstream, it begins to spread everywhere blood flows. Some organs, however, are hit harder and sooner because of the way circulation works. Among the first to encounter alcohol is the liver. This organ functions as the body's primary filter, processing toxins and breaking down substances that could be harmful if allowed to accumulate. In the case of alcohol, the liver begins its work almost immediately. Specialized enzymes are employed to convert ethanol into other molecules through a two-step process. In the first stage, an enzyme called alcohol dehydrogenase, often abbreviated ADH, transforms ethanol into acetaldehyde. Acetaldehyde is highly reactive and more toxic than ethanol itself. If it were allowed to linger in the body, it would cause severe damage. To prevent this, a second enzyme, aldehyde dehydrogenase, or ALDH, quickly converts acetaldehyde into acetate, a much less harmful compound. Acetate can then be further broken down into carbon dioxide and water, substances the body can easily eliminate. The liver's detoxifying role is continuous. As long as alcohol is present in the bloodstream, the liver works steadily to reduce its levels. However, the liver can only process a fixed amount at a time. Excess alcohol that has not yet been metabolized continues to circulate, reaching other organs. The brain, supplied by rich blood flow, becomes one of the most important targets. The degree to which alcohol manages to escape liver breakdown during this first pass strongly determines the intensity of intoxication, because it dictates how much ethanol actually reaches the brain's neurons. When ethanol molecules penetrate the brain, they begin altering its normal patterns of communication. The brain operates through a balance of chemical signals. Some signals accelerate activity, pushing neurons to fire and relay information. Others slow activity, holding neurons back and providing stability. Two of the most important messengers in this system are glutamate, which acts like an accelerator, and gamma-aminobutyric acid, or GABA, which acts like a brake. Alcohol disrupts this delicate balance by enhancing GABA's calming influence, while simultaneously weakening glutamate's excitatory push. The effect is profound. Neurons become quieter, messages travel less efficiently, and overall brain activity slows. At low to moderate doses, this dampening of activity is felt as relaxation, reduced anxiety, and mild sedation. With higher consumption, the slowdown intensifies, leading to drowsiness, poor coordination, and slurred speech. If enough alcohol is taken in, the suppression of vital brain functions can become dangerous, even life-threatening. The brain's response to alcohol, however, is not only about sedation. Another key effect arises from alcohol's influence on the brain's reward system. A cluster of neurons in the midbrain projects to an area called the nucleus accumbens, a region deeply involved in motivation and pleasure. Ethanol stimulates these pathways, triggering the release of dopamine, a neurotransmitter strongly tied to reward and reinforcement. The surge of dopamine produces feelings of enjoyment and satisfaction, reinforcing the desire to continue drinking. In this way, alcohol shares a common pathway with many addictive substances, all of which hijack the brain's reward machinery to generate pleasure. Alongside dopamine, alcohol also prompts the release of endorphins. These are the brain's natural painkillers and mood elevators, normally produced to help humans endure stress or injury. When ethanol causes endorphin levels to rise, the result is an additional wave of relaxation and euphoria. Together, dopamine and endorphins provide the positive, emotional core of intoxication, encouraging repeated use. Yet, just as alcohol's entrance into the brain brings about intoxication, its gradual removal reverses the process. As the liver continues to metabolize ethanol into harmless byproducts, blood levels fall. Neurons slowly return to their normal pace, and the psychological haze of drunkenness dissipates. This fading effect varies widely across individuals. Some people recover quickly, while others feel groggy or unwell for much longer. The differences arise from a combination of biological and genetic factors. For example, men and women who weigh the same and drink identical amounts under the same conditions can still experience different blood alcohol concentrations. One reason is that body composition differs between the sexes. On average, women have proportionally more body fat and less total water compared to men. Because alcohol dissolves more readily in water than in fat, a smaller volume of blood is available to dilute it in women. This results in a higher concentration of ethanol in the bloodstream after drinking. Thus, drink for drink, women often feel the effects more strongly. Beyond body composition, genetics influence how enzymes in the liver handle alcohol. Variations in the genes encoding ADH and ALDH enzymes can speed up or slow down the conversion of ethanol to acetaldehyde and then to acetate. A person with a particularly efficient version of these enzymes may metabolize alcohol quickly, experiencing less intense intoxication. Conversely, someone with slower enzymes may accumulate acetaldehyde or ethanol, amplifying their effects. Repeated drinking adds another layer of complexity. Regular exposure to alcohol can stimulate the liver to produce more of the enzymes responsible for its breakdown. As enzyme levels rise, the liver processes ethanol more rapidly. This contributes to tolerance, where increasing amounts of alcohol are needed to achieve the same subjective effects. But the process is not limitless. In individuals who drink heavily over long periods, the liver can become damaged. Scar tissue, inflammation, or cirrhosis may impair the organ's ability to produce enzymes effectively. Instead of increasing tolerance, liver damage slows metabolism, causing alcohol to remain in the bloodstream longer and heightening its toxic impact. The variability does not end at metabolism. The brain itself contains genetic differences in how it responds to neurotransmitters. Some individuals naturally have lower baseline levels of dopamine or endorphins. For them, alcohol-induced boosts in these chemicals may feel especially rewarding, increasing the risk of repeated use as a form of self-medication. Others may carry variants of genes involved in GABA signaling, making them particularly sensitive to alcohol's sedative effects. These individuals might be less likely to develop dependence because drinking rapidly makes them sleepy rather than euphoric. Such differences show why the same dose of alcohol can generate pleasure in one person and lethargy in another. Over time, persistent alcohol use reshapes the brain's internal balance. Neurons adapt to the constant presence of ethanol by adjusting their own chemistry. GABA signaling becomes less responsive, dopamine and endorphin activity diminish, and glutamate, the excitatory counterpart, grows stronger. The result is a nervous system that feels underactive without alcohol, but overstimulated with it. This adaptation explains why chronic drinkers often feel anxious, restless, or unable to sleep when sober. What once felt like normal functioning now feels uncomfortable, pushing them to drink again to restore temporary balance. This cycle is one of the hallmarks of alcohol use disorder. Understanding alcohol's effects, then, requires tracing its journey from the moment it is swallowed through its chemical processing in the liver, its disruption of neural circuits in the brain, and its eventual clearance. Every stage is influenced by a mix of biological sex, genetic inheritance, metabolic efficiency, and history of prior consumption. Together, these factors shape the unique experience of intoxication for each individual. For some, alcohol remains an occasional indulgence that relaxes the mind. For others, it becomes a repeating pattern that rewires the brain and body, locking them into cycles of dependence. When considering why alcohol has remained such a persistent presence in human life, it is not just its cultural or social significance that matters, but also the complex biological processes that determine how each sip influences the body and the mind. Ethanol may appear to be a tiny chemical, but once it is consumed, the way it interacts with living tissue reveals an intricate story of chemistry, physiology, and neurobiology. To fully understand the wide spectrum of drunken states, it helps to look at what is happening inside, at levels invisible to the naked eye, but fundamental to our experiences. The very simplicity of alcohol's chemical structure explains its remarkable versatility in infiltrating human systems. Molecules of ethanol are small, light, and not heavily burdened by large groups of atoms. Because of this, they can slip through cell membranes with little resistance. In practical terms, that means once alcohol enters the digestive system, very little prevents it from quickly crossing into the bloodstream. Larger, more complicated compounds often need transport proteins or lengthy processes before they can get into circulation. Ethanol does not. It simply passes through weaving into blood plasma within minutes of ingestion. From this point onward, blood circulation becomes the vehicle carrying ethanol throughout the body. Blood is pumped by the heart to every corner, ensuring that alcohol's effects are not isolated but widespread. Areas with particularly rich blood supply, such as the liver, lungs, and brain, are especially affected. The liver, being the primary hub for detoxification, continues its relentless task of converting ethanol into byproducts. However, since the liver's capacity is limited, alcohol molecules that escape immediate processing remain in circulation, free to interact with other tissues. The brain, with its dense network of blood vessels and its hunger for oxygen and nutrients, becomes one of the earliest and most heavily influenced targets. The uniqueness of alcohol's action lies in how it interferes with communication among nerve cells. Neurons are designed to send electrical impulses, but they do not function in isolation. Chemical messengers called neurotransmitters bridge the gaps between them. When alcohol seeps into this environment, it alters the balance of these messengers. It boosts the influence of gamma-aminobutyric acid, the neurotransmitter that applies the brakes, while diminishing the strength of glutamate, which normally pushes the gas pedal. This dual action of pressing harder on inhibition and easing up on excitation leaves neurons far less active than they would otherwise be. The result for the drinker is noticeable in behavior. Relaxation, slowed reaction times, dulled reflexes, and sometimes a warm sense of calm. Yet, if consumption continues and blood levels climb higher, the dampening of brain activity deepens. Slurred words, poor balance, loss of coordination, and eventual unconsciousness can follow. In extreme cases, the suppression of brain regions controlling vital functions, such as breathing or heart rhythm, can be fatal. The pleasure-producing side of alcohol is equally tied to its manipulation of brain chemistry. Within the reward system, the presence of ethanol stimulates dopamine release. This neurotransmitter, associated with the brain's reward prediction system, makes the act of drinking feel satisfying and reinforces the behavior. The nucleus accumbens, a critical hub for motivational processes, lights up with dopamine activity when alcohol is present. This is the same neural circuitry exploited by many addictive substances, from nicotine to opioids. But alcohol has an added dimension. It also promotes the release of endorphins, natural opioids produced within the brain. Endorphins not only relieve discomfort, but also elevate mood, creating a sense of well-being that amplifies the pleasant aspects of drinking. This combination of dopamine and endorphins forms the emotional core of intoxication. The feelings of pleasure, relaxation, and sometimes euphoria that drinkers often seek. Even as these rewarding sensations occur, the liver is working steadily to bring alcohol levels down. Metabolism of ethanol follows a relatively constant pace, dictated by enzyme activity rather than by willpower. On average, the liver can handle roughly 1 standard drink per hour, though this varies among individuals. Because this rate cannot be accelerated significantly, once a person consumes more alcohol than the liver can keep up with, the excess remains in circulation, extending intoxication until enough time has passed for metabolism to catch up. This explains why attempts to sober up quickly, whether by drinking coffee, exercising, or splashing cold water on the face, do not actually remove alcohol from the blood. Only the slow, steady work of enzymes can accomplish that. Yet, even though metabolism follows predictable rules, the way alcohol feels can be vastly different from one person to the next. Sex, weight, genetic background, and drinking history all influence blood alcohol concentration and neural sensitivity. Women, because of lower average body water content and higher fat proportion, typically reach higher concentrations with the same intake as men. Genetics add another layer. Variations in the genes coding for ADH and ALDH enzymes determine how quickly ethanol and acetaldehyde are processed. Some populations carry versions of these enzymes that metabolize ethanol very efficiently, but convert acetaldehyde slowly, leading to uncomfortable flushing and nausea when alcohol is consumed. Others have the opposite, producing tolerance more quickly. These inherited differences partly explain why susceptibility to alcohol dependence varies among groups and why individuals have such unique responses. Tolerance itself is a story of adaptation. The nervous system, when repeatedly exposed to alcohol, does not remain static. Neurons adjust their receptor levels, neurotransmitter output, and overall excitability in response to the continual dampening effect of ethanol. Over time, this leads to a brain that has recalibrated itself. When alcohol is present, balance feels normal. When it is absent, the altered brain chemistry produces restlessness, anxiety, irritability, and sleep problems. This is the paradox of chronic alcohol use. The very substance once used for relaxation and pleasure becomes necessary simply to feel ordinary. Without it, the system feels unstable, creating a powerful drive to drink again. This shift marks the threshold between occasional use and dependency. The risks extend further when prolonged consumption injures the liver itself. Chronic exposure to high alcohol levels can scar the organ, inflame tissues, and destroy functional cells. With fewer healthy liver cells available, enzyme production drops, reducing the organ's ability to clear alcohol efficiently. This means intoxication lasts longer, blood levels rise higher, and damage to other systems accelerates. At the same time, toxins accumulate, compounding harm. What once felt like tolerance eventually gives way to heightened sensitivity. But in the context of a failing liver, this sensitivity is dangerous rather than protective. Another striking aspect of alcohol's variability lies in psychological response. Some individuals, due to lower baseline dopamine or endorphin activity, may experience a dramatic mood lift when drinking. For them, alcohol can temporarily fill a neurochemical gap, making social interactions easier or emotions lighter. This relief, however, comes at a cost. It makes repeated drinking more tempting. Others with naturally high sensitivity to GABA's sedative effects might experience more drowsiness than pleasure, which can discourage frequent use. These differences are rooted in the underlying wiring of the nervous system, sculpted by both genes and past experiences. No two brains are identical, and no two responses to alcohol are exactly the same. As alcohol's influence fades when liver metabolism catches up, the after effects can vary as well. Some individuals wake clear-headed once ethanol levels decline, while others experience headaches, dehydration, fatigue, and nausea, symptoms collectively known as a hangover. The causes are multifaceted: acetaldehyde accumulation, dehydration from alcohol's diuretic effect, and immune system responses all contribute. Just as with intoxication, the severity of after effects differs widely across people, reinforcing the theme that alcohol's journey is personal as much as it is biological. Stepping back, it becomes clear that drunkenness is not a single state, but a continuum shaped by dosage, physiology, and individual brain chemistry. One drink may cause a mild shift in mood and social ease, Several drinks may slow coordination and cloud judgment. Larger quantities risk shutting down essential brain centers. At every stage, personal differences in body composition, enzyme activity, neural sensitivity, and prior exposure alter the exact trajectory. This complexity helps explain why alcohol, though chemically simple, produces such varied and sometimes unpredictable outcomes. It is not just the molecule itself, but the unique human environment it enters that determines the experience. The consequences of this variability are profound. Social drinking, when modest, may produce short-term relaxation without lasting harm. But heavy or chronic consumption reshapes biology in ways that persist long after the last glass is emptied. The nervous system learns to expect ethanol, the liver adapts or deteriorates, and psychological patterns develop around the presence or absence of alcohol. Over years, these changes can transform drinking from a choice into a compulsion, illustrating how a small molecule can wield outsized influence over human lives. Ethanol's ability to influence both body and mind cannot be separated from the way humans are built. The molecule is the same in every glass, but the systems it enters are never identical. Even within a single individual, the context of drinking, time of day, fullness of the stomach, hydration levels, stress state, or fatigue can change the experience dramatically. This flexibility makes alcohol unique among psychoactive substances. A drug with such a simple structure could easily be expected to behave in predictable ways. Yet, its small size and wide reach allow it to slip into countless niches of the human body, producing a spectrum of effects that range from pleasurable calm to dangerous suppression of life-sustaining functions. To explore further, we need to consider in detail how the body handles each stage of ethanol's presence, not only in terms of chemistry, but also in terms of lived human biology. Once swallowed, alcohol begins a journey defined by absorption, distribution, metabolism, and elimination. Absorption varies enormously depending on circumstances. When the stomach is empty, ethanol passes quickly into the small intestine, whose folded lining provides an enormous surface area lined with blood vessels. This means alcohol enters circulation rapidly, raising blood alcohol concentration within minutes. On the other hand, when food fills the stomach, digestion slows, and alcohol remains trapped behind the pyloric sphincter. The longer it stays there, the slower it reaches the small intestine, reducing how much ends up in circulation at once. This is why the same drink might feel mild after dinner, but potent before eating. The biology of digestion, often unnoticed, profoundly shapes the intoxication curve. Once absorbed, ethanol disperses into water-rich compartments of the body. Because the body is composed of varying proportions of water, fat, muscle, and bone, the distribution is different from one person to another. Muscle tissue, rich in water, provides a larger reservoir for dilution than fat, which contains relatively little. Men typically have more lean muscle and therefore more total water, giving ethanol a larger volume to spread through. Women, with higher relative fat content and less water, concentrate alcohol into a smaller space, leading to higher blood alcohol levels for the same dose. These biological differences are not trivial. They explain why sex-based comparisons of drinking can be misleading without accounting for body composition. The blood, carrying ethanol, passes first through the liver before circulating widely. The liver acts as the gatekeeper, beginning the breakdown process as soon as molecules arrive. Alcohol dehydrogenase converts ethanol into acetaldehyde, a compound far more reactive than its precursor. Acetaldehyde is toxic, capable of causing cell damage and unpleasant sensations if it accumulates. Fortunately, aldehyde dehydrogenase steps in to convert it into acetate, a molecule that can be processed into carbon dioxide and water. But the efficiency of this sequence is not the same in everyone. Genetic variations mean that in some individuals, acetaldehyde lingers longer. This leads to facial flushing, rapid heartbeat, nausea, and dizziness after even modest consumption. In others, enzymes work quickly, lowering discomfort but potentially increasing risk of overconsumption because the body's warning signals are less pronounced. These inherited traits underscore how biology predetermines much of the alcohol experience. While the liver quietly processes molecules, ethanol continues to circulate, reaching the brain where its signature effects occur. Within the brain, neurons communicate via synapses, tiny gaps bridged by neurotransmitters. Alcohol tilts this chemical conversation. By amplifying GABA, the neurotransmitter responsible for calming and inhibiting neural firing, and simultaneously muting glutamate, which normally excites and activates, ethanol slows the tempo of brain activity. Speech becomes harder to articulate, reaction times lengthen, movements grow less precise. The drunk state emerges not from one single action, but from a broad dampening of communication across brain circuits. At low levels, the effect feels like relaxation and lowered anxiety. At higher concentrations, the same mechanisms make standing, walking, and speaking more difficult, showing how a small shift in chemistry can cascade into visible changes in behavior. But alcohol does not only inhibit, it also excites the pleasure system. Deep in the midbrain, dopamine-producing neurons project to the nucleus accumbens, the brain's reward hub. When alcohol stimulates this pathway, dopamine surges, creating a sense of reward and reinforcing the behavior of drinking. Endorphins, naturally produced opioids in the brain, also rise with alcohol intake, further enhancing feelings of comfort and mild euphoria. These neurochemical boosts encourage repetition. Just as food and social interactions reward survival behaviors, alcohol rewards itself, pushing the user toward continued consumption. This reinforcement is one reason why alcohol has such a powerful hold across cultures and centuries. It hijacks systems evolved to encourage beneficial behaviors. The danger lies in the narrow line between reward and harm. As ethanol levels climb, sedation deepens. At very high concentrations, the depressant effect can silence brain regions that control breathing and heartbeat, creating a medical emergency. Unlike some other drugs, alcohol has no clear ceiling where the body simply refuses to absorb more. The same digestive pathways keep working, and unless the drinking stops, levels can climb until critical functions shut down. This duality, pleasure at moderate doses, danger at high doses, explains much of alcohol's paradoxical place in society. Celebrated in moderation, feared in excess. Beyond immediate intoxication, alcohol's journey through the body involves longer-term adaptations. Regular exposure encourages the liver to increase enzyme production. Alcohol dehydrogenase levels rise, making the body more efficient at clearing ethanol. This produces tolerance, where more drinks are required to achieve the same level of intoxication. But tolerance carries a hidden cost. The same adaptations that reduce acute effects also generate strain on the liver, increasing the risk of inflammation and scarring. With prolonged heavy use, liver cells are replaced by fibrous tissue, diminishing function. At that point, alcohol lingers longer in the blood, intensifying toxicity rather than blunting it. Tolerance may seem protective, but in reality, it paves the way toward organ damage. The brain adapts as well. Neurons exposed repeatedly to alcohol change their receptor availability and neurotransmitter output. GABA receptors may become less responsive, while glutamate receptors become more active, compensating for alcohol's constant dampening. Dopamine release in the reward circuit diminishes over time, making natural pleasures less satisfying. Endorphin production decreases, reducing the brain's own capacity to regulate mood. These changes make sobriety feel uncomfortable, characterized by restlessness, irritability, insomnia, and anxiety. Drinking again temporarily restores balance, but the relief comes at the price of reinforcing dependence. This cycle illustrates how alcohol transitions from a source of pleasure to a source of necessity, driving individuals toward compulsive use. Differences among people are visible not only in how they metabolize alcohol, but also in how their brains respond. Those with lower natural dopamine or endorphin activity may find alcohol's boost especially rewarding, making them prone to repeated use. Others with highly sensitive GABA systems may experience more sedation than reward, limiting their consumption. These underlying biological differences contribute to risk profiles for alcohol use disorder. The same drink is not the same experience for everyone. It is filtered through the unique lens of each person's neurochemistry. Cultural context interacts with biology as well. Social norms influence when, how, and why people drink. In some societies, alcohol is tightly woven into rituals and traditions, encouraging moderate, regular use. In others, it is consumed in heavy bouts, increasing risk of harm. But beneath these surface behaviors, the same biology operates. Enzymes, neurotransmitters, and receptors respond in predictable patterns, shaped by genetics, sex, body composition, and history. Understanding these processes clarifies why no two people ever experience alcohol in precisely the same way, and why patterns of use vary so widely. Hangovers provide another glimpse into alcohol's systemic impact. After blood levels drop and intoxication fades, the body continues to feel consequences. Dehydration, caused by alcohol's diuretic effect, leaves tissues parched, producing headaches and fatigue. Acetaldehyde, if it accumulates, contributes nausea and malaise. The immune system may become activated, releasing molecules that cause inflammation and amplify discomfort. These combined factors explain why some people feel barely affected the morning after, while others suffer prolonged symptoms. Just as intoxication differs, so does recovery, shaped by metabolism, hydration, and individual sensitivity. Ultimately, alcohol's story in the human body is one of interaction between molecule and enzyme, neuron and neurotransmitter, culture and biology. Ethanol itself is unchanging, yet its effects are endlessly variable because of the diverse human systems it enters. It slows, it rewards, it sedates, it excites, and it harms, depending on dose, context, and individual traits. This complexity is what makes alcohol both fascinating and perilous. A single glass can relax or destroy, depending on who drinks it, how much, and under what circumstances. Tracing its path through the body shows how deeply it entangles itself with human biology, altering not just momentary states of mind, but long-term patterns of health and behavior. The journey of alcohol within the human body is not just a short tale of ingestion, absorption, and elimination. It is also the story of how repeated exposure can reshape organs, restructure brain chemistry, and even modify behavior itself. Ethanol is small, but its influence is expansive. The cascade of events that follows each drink is not isolated to one evening. It stretches forward into the next day, the following week, and in the case of regular use, across years of life. To truly grasp the full impact of this compound, one must consider not only the immediate intoxication, but also the enduring imprints it leaves upon human biology and psychology. When alcohol enters the bloodstream and eventually reaches the brain, the primary effect is sedation through the manipulation of inhibitory and excitatory neurotransmitters. But the mind is not just subdued. It is also tricked into perceiving reward. Dopamine and endorphins surge, convincing the brain that something good has happened. This illusion of pleasure is powerful because it teaches the brain to connect alcohol with relief, sociability, or comfort. Over time, this link can grow so strong that the mere expectation of drinking can activate reward circuits, creating craving even before the first sip. The association between alcohol and reward is not a conscious decision, but a deeply embedded neural adaptation. The body learns, and once learned, the pattern is hard to unlearn. Yet this pleasurable reinforcement is balanced by the body's natural drive for equilibrium. With repeated alcohol exposure, the brain begins to anticipate sedation and reward. To counteract, it reduces GABA sensitivity, increases glutamate signaling, and tempers dopamine release. In this altered state, alcohol is no longer just a source of enjoyment. It becomes a necessity for feeling normal. Without it, the brain tips into imbalance, producing anxiety, sleeplessness, irritability, and restlessness. What was once optional becomes obligatory. This transition illustrates the progression from casual drinking to dependency. The user may feel as though alcohol provides relief from discomfort, when in reality, the discomfort exists because of alcohol's long-term presence and the adaptations it forced upon the nervous system. The liver follows a similar trajectory. In the early stages of drinking, enzyme production ramps up, increasing the speed of ethanol metabolism. This can give the illusion of resilience, as the same number of drinks produce milder effects. But this tolerance is deceptive. It signals that the liver is under strain, constantly being pushed to work beyond its usual capacity. Over months or years, cells die, scar tissue forms, and inflammation spreads. Eventually, the organ's efficiency declines, leading to slower alcohol clearance and accumulation of toxins. Instead of protecting the body, tolerance flips into vulnerability. The once resilient drinker now becomes more susceptible to harm because the organ designed to protect them has been damaged. The consequences are not limited to the liver. Alcohol in circulation touches every system. The heart must pump thicker, more viscous blood when alcohol dehydrates tissues. The pancreas may become inflamed, disrupting digestion. The immune system can weaken, leaving the body more susceptible to infections. The stomach lining can erode, leading to ulcers. Every organ that comes into contact with alcohol's journey is affected in some way. And the cumulative impact is far greater than the sum of each individual consequence. The brain may crave alcohol's presence, but the rest of the body pays a heavy price for accommodating it. Individual variation continues to shape this journey at every step. Two people can drink the same amount and display entirely different responses, not just in intoxication, but in long-term outcomes. One individual may develop tolerance rapidly. Another may remain sensitive for years. Some may metabolize acetaldehyde quickly, sparing themselves from flushing but inadvertently increasing risk of addiction. Others may suffer immediate discomfort from even small amounts, deterring heavy use but causing social challenges. Genetics, body composition, health status, and drinking history all intertwine to make alcohol's path through the body uniquely personal. This is why no universal measure of drinking can fully predict outcomes for every person. Behind every glass of alcohol lies a complex interplay between molecule and biology, one that is repeated in countless variations across the population. Hangovers provide a smaller, but still revealing glimpse of this interplay. After intoxication fades, the body wrestles with the aftermath. Dehydration caused by alcohol's interference with antidiuretic hormone leaves tissues craving water. Acetaldehyde accumulation irritates tissues and unsettles the stomach. Immune system responses release molecules that contribute to fatigue and malaise. Sleep, though it may come easily after drinking, is shallow and fragmented, leaving the brain unrested. The combined effect is a reminder that alcohol's impact is not confined to the period of intoxication. It lingers, reshaping the body's balance long after ethanol has left the bloodstream. Some people bounce back quickly, others suffer prolonged symptoms, but the underlying cause is the same: alcohol's broad disruption of physiology. Chronic use compounds these disruptions into structural changes. Neurons reorganize their synapses, receptors are upregulated or downregulated, and signaling pathways shift. The reward system, once easily activated by small amounts of dopamine, becomes dulled, requiring more stimulation for the same effect. Natural pleasures, exercise, music, companionship, lose their shine compared to alcohol's artificial boost. The individual, seeking to recapture lost feelings of pleasure, drinks more, yet the response diminishes with each attempt. This is the vicious cycle of alcohol use disorder. The pursuit of a reward that grows increasingly out of reach, coupled with withdrawal symptoms that make abstinence painful. From a biological standpoint, dependence is not a moral failing, but a predictable outcome of repeated chemical interference with brain function. Ethanol forces the brain into states it would not naturally occupy. And the brain, in turn, adapts to survive under those conditions. The adaptations themselves create discomfort when alcohol is removed, leading to continued use. This cycle explains why some individuals, even when fully aware of alcohol's harms, struggle to stop. Their brains and bodies have been rewired to expect alcohol's presence, and absence feels intolerable. Despite these risks, alcohol's role in human culture has been remarkably enduring. Across civilizations and centuries, it has been celebrated in rituals, feasts, and social gatherings. Its immediate effects, relaxation, reduced inhibition, enhanced sociability, have made it a tool for connection and expression. But beneath these cultural layers, the biology remains the same. Ethanol sedates, rewards, and adapts the body, regardless of whether it is consumed in a grand banquet or in solitude. Understanding this duality, cultural celebration versus biological consequence, is key to seeing alcohol for what it truly is: a chemical that reshapes human experience by altering the most fundamental processes of the body. The long-term story of alcohol also includes resilience and variation. Some individuals can drink moderately throughout life without ever developing dependency. Their enzyme levels, receptor sensitivities, and cultural habits align in ways that keep consumption from spiraling into disorder. Others, with less favorable genetics or higher stress levels, may find themselves pulled into problematic patterns even with modest use. The diversity of outcomes reinforces the idea that alcohol is not a uniform experience. It is a shared molecule, but it tells a different story in every body it enters. Eventually, as ethanol is metabolized fully into carbon dioxide and water, it leaves the body. But the memory of its presence remains, in the altered receptor sensitivity of neurons, in the strained function of the liver, in the reinforced pathways of the brain's reward circuits. These echoes are why tolerance develops, why dependence takes root, and why recovery can be difficult. Abstaining does not instantly reset the system. It requires time for the brain to rebuild balance, for the liver to heal, for the body to relearn normal function without alcohol. This recovery process is possible, but it demands patience and persistence, because the adaptations caused by ethanol do not vanish overnight. Ultimately, alcohol's path through the body is a story of contrast. It is a chemical both small and powerful, both rewarding and destructive, both socially celebrated and biologically costly. Its effects unfold in stages: rapid absorption, systemic distribution, partial detoxification, neural interference, rewarding stimulation, lingering after effects, and eventual elimination. At each stage, personal biology dictates outcome, making every drinking experience unique. Yet across these differences, the same principles apply. Alcohol slows, it rewards, it adapts, and it harms. The more often it is consumed, the more deeply it entangles itself in the fabric of human physiology. Ethanol's simplicity belies its complexity of effect. It is not just a molecule, but a catalyst that reshapes the systems it touches. By understanding how it travels, how it is processed, how it manipulates the brain, and how it leaves behind traces of adaptation, we see clearly why alcohol has both captivated and troubled humanity for millennia. Its power lies not in its size, but in its ability to interact broadly and persistently with the machinery of life. And as long as it remains present in human culture, the story of ethanol will continue to be written in the chemistry of the body and the behavior of the mind.